Okay, welcome. Good afternoon, everybody. For some people, it might be not good afternoon, but good morning or good night. My name is Michiel Wels. I will be the person responsible for presenting this webinar today to you about bioinformatics. First of all, I would like to welcome you all and to the first NIZO webinar that was ever given by us. So don't be afraid, we checked everything, everything will be all right, but just to show you, you are a witness of this first BioIT webinar. So today we will discuss a few topics, and before I will introduce this, I want to introduce to you my colleague Jos Boekhorst, who is sitting next to me here in the room. He will be the main person responsible for addressing the questions, and I would like to give him the word now to explain a few of the household things that need to be discussed before we start with the content. Jos, the floor is yours.
Thank you, Michiel. So, Jos Boekhorst, I'm also a project manager Bioinformatics at NIZO, and this afternoon I will be assisting Michiel by answering any questions that come in at my machine over the chat function of the webinar software. So, on the menu on the right side of the screen there is a chat, and if you type any questions in there, they will show up only at my machine, and I will try to answer all your questions. So don't hesitate to type anything you feel like. If there are any questions that I think are urgent to answer plenary by Michiel, I will interrupt Michiel and ask him to address things that might be unclear in the presentation itself, but I will try to answer most of your questions also by text. If it is very busy with many questions, or if I think questions basically desire a more elaborate answer than I can quickly type now, we will get back to you at the end of the webinar or by email if I think we need other input from other people at NIZO. Still, don't hesitate to type anything you'd like to know during the presentation. So I can either interrupt Michiel, or at the end of the webinar we can also discuss stuff plenary if necessary. If you have problems with your audio, you can choose to use a phone to get audio, or use your PC or your laptop. You can also change that on the right side of the screen in the audio tab of the webinar software. Okay, Michiel, I think that's the most important stuff. So the floor is yours.
Okay, thank you very much, Jos. So let's start with the webinar. What I wanted to talk to you about today is bioinformatics, and the reason I think we should discuss this in a webinar like this is that I think that if you look at food and food research, microbes are key players in that. And of course microbes are known for many different, to have many different functions and display different functions in food products. First of all, you have of course microbes, which one example of that would be Lactococcus lactis, which you can use in fermented products, which basically take your basal raw material and make something really nice out of it, like in this case cheese. On the other hand, microbes are more and more used within the food research as adjuncts that can have, for instance, a health benefit. When you think about, for instance, probiotics, and then an organism like Lactobacillus plantarum is a nice example of a microbe which is used to enhance the health of your product. And finally, of course, there's also the downside of having microbes in your food products, and those are related to food spoilage, where microbes spoil your food, and you would like to control these microbes as well so that they don't display a clear function in your product. And what I want to discuss with you today is that apart from all the classical techniques of studying these microbes, genome sequence information and genomics will help you to enhance the insight on how these microbes contribute to your product. And if done properly, it will give you a real mechanistic view on how to improve your product. So the main topic of today is basically based on genomics. Genomics was is a name for basically all studies of organisms that relate to the genome and the function of their genes. So it can either be just looking at a genome of an organism, looking at a transcriptome, so the genes, how are they described, but also on more on a protein level, that you could call proteomics. Genomics is known to really give you a high quality and broad overview of the complete organism, so you can get a study of all the genes which are in your organism. And of course this gives you a lot of data, but then of course the question is, how can you use that data, which is basically a rich source of information, to help you improve your strains or improve your product? And I think the answer of the webinar of today, and I hope to convince you at the end of this webinar, that you will really need dedicated bioinformatics to get from just having a bunch of genes which tell you something about the organism all the way down to improving strains and improving processes. Okay, because we cannot really cover all topics of bioinformatics today, I want to limit myself to 3 different topics. The first thing I want to show you today is our analysis that are related to genome sequencing and the analysis of a genome sequence, more specifically looking at annotation of a genome and taking that a step further in comparing different genomes and perform a matching of the phenotype to the genotype. Secondly, I want to discuss with you something which we call metabolic modeling, where we use the genome sequence to build a genome scale model of all metabolic processes that go on in the cell, and how this can help you, for instance, in visualizing and interpreting your transcriptome data, but also how you can use this to really perform modeling, so flux, balance, derived analysis that makes it able to predict the growth of your organism by means of the genome sequence. And finally, the last topic of today will deal with basically the development of microbial community profiling techniques such as barcoded pyrrole sequencing, which helps you to look at communities which can be available close to the human body, but also all kinds of environmental samples, and how you can identify key players, so the main important microbes during, and how these are affected when you put a certain treatment on these organisms. Okay, so starting off with our first topic, genome sequence data analysis. So genome sequencing and annotation is now already a well-established technique, even up before 2000, before the year 2000, people started sequencing their microbial genomes. Drawback back then was still that the techniques that we used for sequencing were very labor-intensive, and sequencing a microbial genome was still very expensive, so over €1 million. Secondly, because of the use of these type of techniques, you really had to spend a lot of money in trying to get the gaps that you would still have in such a genome closed by dedicated tests like multiplex PCR and manual PCR. After that, because there was such a limited database with annotated genome sequences, annotation was also still a very time-consuming process. So annotation would still take more than a man's year of work, and it will end up in, well, a limited knowledge of your organism, because you could not compare it enough to already present and available genome sequences. Well, after 2005, with the introduction of high-throughput sequencing methods, sequencing really dropped in price. So you would really go down from €1 million all the way down to only €100,000 to sequence an organism. This made it available for many different people, if they had a model organism that they wanted to study, to go and sequence that. In addition, because new techniques were invented also to do the gap closure was also also went very much faster than that. And of course, because all kinds of people started annotating their genomes, annotation would take far and less time, and because of the availability of more and more genomes, it would become much cheaper. Finally, I think after the year of 2009, with the development of even better high-throughput sequencing methods, sequencing became even more cheaper. So nowadays, if you want to sequence a genome, this costs around €6,000 up up until down to 500 euros, depending on what you want to know from such a genome. With the development of these methods, also new approaches in how to sequence these organisms required only a limited number of gap sequencing, so that gap closure was also becoming more and more attractive and more faster to do. And even nowadays, most of the people don't even want to close their gaps anymore because they contain only very little information that you would miss. Finally, annotation has also improved even more because nowadays bioinformatics groups all over the world have developed some kind of semi-automated annotation pipelines that you can just run when you have a unannotated genome. You just put it into a website, the genome is annotated, and after a few days you get a completely annotated genome back. However, these annotation pipelines still contain a lot of errors, which are just introduced by errors that are already present in databases and that are just copied to your new genome. And just to give you an example of a few of these annotation pipelines, you can see them here. Most of them developed by universities, freely available for you to submit your genome sequence and download the end result, which is a complete annotation of your genome. Now, because these annotation pipelines also still contain a lot of errors, you might think that maybe having such a single annotation pipeline do the work for you is not the best option. So what we developed here at NIZO, and which shown to be pretty successful already, is that we've built a pipeline that allows you to compare all the different results from these different automated annotation pipelines and basically builds up some kind of a consensus alignment. So what it does, it takes the results of these different annotation pipelines, combines them into a single annotation, which you can then download, you can look at, and of course this then is your starting point for manually annotating your genomes. So basically how this system works is that you take a knowledge base, so genome annotations that you've performed in the past, of which you know that they are of high quality, and we have several of those here at NIZO, like the genome of Lactobacillus plantarum WCFS1 or the genome of Lactococcus lactis KF147, and those are high quality. So if you can get the quality out of that annotation, you don't really need to redo the full curation process that you've undergone with those already known, already annotated organisms. If you then have a new genome, basically what you do is you add that genome to the pipeline, you compare it, you perform the annotation, and on basis of the quality rules and the translation lists that you have of these previously annotated genomes, you basically distill all the information from the different pipelines and combine them into a new one. And basically, and by each time adding a new genome to this database, of which you also know that it is now high quality annotated, the rules in which you apply to get this completely annotated genome become better and better. And well, to our experience, this is a very useful tool to get a high quality prediction based on automated procedures, so that they don't take you too much time, and it reduces the number of genes that you have to look at by eyeballing, so by scanning them manually, is about a reduction of 50%. So 50% of the genes that you predict are now okay, and you don't need to look at them anymore by hand. Of course, for the rest of the genes that you could not annotate in this way, of course, there's still the need to manually curate them. So basically what I tried to show you in these first few slides here is, first of all, sequencing has become very cheap, so it's only a few €100 to get a genome sequenced, and also the downstream analysis, so going from a genome that has been assembled up and down to a genome that is completely annotated is no longer a bottleneck anymore. So basically this allows you to just take a big collection of strains, which you use in your factory, for example, and just send them for sequencing and annotation. Now, how can this then be useful? Now, it's especially useful because you can now take these genomes, for which you know that these have all kinds of different properties, and you can just analyze them on, well, what do they contain, and why do some of the strains display a certain property while others do not. For example, you can take your genomes now and you can mine them for the presence of all kinds of interesting factors or maybe unwanted factors in your genomes. So if you're interested in finding all kinds of new metabolic features, you can now just scan the genomes to see whether the genes that display this specific metabolic feature are there, because those are the likely candidates that will allow you to produce this specific metabolite, for instance. In addition, looking at the safety of your genome, you can also look at what's the presence of, for instance, antibiotic marker genes, or other virulence factors in my genome, things you don't want in the genome, and on basis of what you can decide maybe to not take this strain into account in your products. The picture I'm showing you right now is a schematic representation of such a genome comparison. What you see here is basically a large plot which is based on, in which you can see the presence and absence of the genes. So every line in this plot represents a single strain, and in addition, the strain is colored, and all the genes organized on the genome are colored here either in white when they're present or in black when they're absent. You can see in the middle there's this one strain that is completely white. This you could call your reference strain. So this is the strain to which you refer all the others. And you can see that this is completely white, so all genes are present. But the next thing you can see is that for this collection of genes, there's also cases where you can see big blocks of black bars that show you regions where there's a lot of variability between the different strains. So these you could call some kind of genome hotspots, where the gene presence and absence is highly variable, and this might be the result of massive gene gain or loss, and can be of a functional importance when comparing the strains. And finally, what you can also do on basis of these plots is you can try to organize the genomes on basis of their presence and absence. So when you look at the right of this graph, you can see there's a tree drawn here, and this basically reflects the relation of the genomes to each other. And this can tell you something about the evolutionary history of the strains, or maybe it tells you something more about the niche in which these strains are found. And just to give you a broader view on this is an example of different Lactobacillus lactis genomes that were compared, and you can see here that there's a tree here, and this tree is showing you the variability of the genes that are found on the chromosome of these strains. And you can see in the top, those are all Lactobacillus lactis subspecies Cremoris strains, and when you look at the bottom of the tree, these are all Lactobacillus lactis subspecies lactis strains. So this really shows you that chromosomal genes sort of show you the relation, the evolutionary history and the relation of these strains to each other in the course of evolution. On the other hand, the second graph I show you here is showing you the same strains organized now differently in a different tree, a tree based on the genes that are present in plasmids. And you can see already that there's no clear distinction anymore between subspecies Cremoris and subspecies lactis strains. But if you look more closely at the tree, and you can look at, and if you look at it better, you can see there's all kinds of small squares, black squares indicating these strains. These black squares indicate the strains that have a specific dairy origin. So they were isolated from dairy products, and you can see that plasmid content is now showing you a better view on the niche distribution of these strains, which also makes sense, of course, because the chance of having a plasmid transferred from one strain to the other when two strains are in the same niche is, of course, far more likely than when they would be in different niches. Okay, so if you have these genomes and if you have a good basis of knowing which genes are present and absent in the different strains, you can also use this for an even more dedicated search in cases where you want to find the function of a gene in relation to a phenotype. And this is what we call gene trait matching. Basically what you want to do is you want to take the genotype data and the phenotype data and merge that together in some kind of an algorithm, and then what comes out of this algorithm would be then genes that are, of which the distribution pattern of these genes is basically matched with the presence or the absence of a certain phenotype. So you want to find genes whose presence or absence enables the strain to have a certain trait. This, of course, you can use, for example, to re-annotate genes. If you don't know the function of a gene, but you find it popping up linked to a specific phenotypic trait, you know that this gene is probably involved in this specific phenotype. And of course you can think of many different applications of this, for example, related to probiotic features. So if you measure the immune response of different strains on the immune response of different strains and you have the genomes to compare it with, you can find genes related to the immune response or, for example, to pathogen inhibition. You can also do this, of course, in relation to spoilage conditions, so outgrowth under specific product conditions, flavor formation, process conditions. Basically anything you can measure on these strains and in which they differ from each other, you can use it as a basis for matching it with the genotype. So how does this in principle work? What we basically do is we take the genes and we treat them as variables, so things that differ between the strains. And our goal is then to have a computer model that recognizes unknown strains and decides whether they grow or not grow, in this case, on this example, on sucrose, on basis of the genes, so on basis of the gene content. So in this case, let's say we would have a few strains for which we know the growth. It's either they grow, the strains on the left, or they don't grow, the strains on the right. And then if we have a new strain, we basically want to put it in the right box. So do we put it on the left or do we put it on the right? On basis of, does it grow or doesn't it grow on sucrose? And we do this only with the genome information without taking into account any phenotypic tests for this specific one strain. Just to cut this short with an example, here you can see an example of such a table where you can see in the rows the different strains, and then all the columns in the table display different genes except for the last column. The last column is showing you the results of the phenotypic test. So is it able to grow? It means it gets a score of 6, or not grow, meaning it gets a score of 0 on sucrose. And as you can see already in the pattern of presence absence of these specific genes, and I did not select them on coincidence, there was a meaning for that. You can see down below are all strains that don't grow, and when you look at the gene content of these specific genes, in most of the strains this gene is absent. While if you took a look at all the strains that grow on this specific sugar, you can see in the top that most of the strains that can grow on sucrose display the presence of this gene. And you can do this also for other phenotypes. This is showing you an example of growth on sugars, on arabinose. And the same you can see here, indicating in green and red, green indicating absence of the gene for this specific phenotype. The other indicates presence of this gene for this specific phenotype. And you can clearly see that there's a block of genes in the middle which glow red and green, meaning absent in one, present in the other, indicate related to the growth on arabinose. And when you take those genes and you look at the genome sequence, this of course all makes sense because the gene cluster you can see above here is a gene cluster related to all kinds of genes that are involved in the in metabolizing arabinose. So this is summarizing the first part of this webinar. What I tried to show you today on basis of genome sequencing and the data analysis of that is that we're moving away from sequencing single strains, and now sequencing of complete strain collections is indeed very much in view of doing. Secondly, there's all kinds of automated annotation processes, and this allows you to really get a fast and accurate gene function prediction. And by combining all these automated annotation processes, you also get a high quality prediction as well. And finally, what I tried to show you here is that comparison of the strains and coupling the genotype to the phenotype really allows for identification of marker genes responsible for a desired phenotype. Okay, so quickly move on to our second topic of today, related to genome scale metabolic modeling. Basically what you want to do in genome scale metabolic modeling is you want to take the genome sequence as a whole and try and build a model that displays all the enzymatic functions of the genes in your genome. And how you can look at this is that basically you take the genome sequence as an inventory of all the enzymes and metabolites that are available in your organism. And basically you could also compare this a bit to having a roadmap. So a roadmap where the enzymes coupled to the genes are the roads and the metabolites are the cities. And the roads, the enzymes, connect the cities to each other. Then of course when you have such a roadmap, so a complete network of the organism, you can basically link all these roads and cities together and to give you a complete inventory or an encyclopedia of your organism. So what is it able to do? Where can I really benefit on having this microbe present in my food product? And finally, and that's the most important, I think the most interesting part of these genome scale metabolic models, you cannot only do qualitative analysis, but if you do it right and if you make it high quality, you can even do quantitative analysis on these models. So you can perform calculations related to, well, where do I predict the traffic jam? So where's the bottleneck in my metabolic model? How can I get fastest from A to B, or how can I get shortest from A to B? So what are the routes that my organism takes throughout its metabolic capacity to basically to grow and to function in a specific environment? Actually, if you look at it that way, these metabolic models are some kind of a GPS navigator or TomTom for bioconversions. This picture shows you an example of such a metabolic model, and in this case we're looking at a reconstructed metabolic model of Lactobacillus plantarum. And what you can see in this picture here in red, so the red dots are all the metabolites which are present in the organism, and the blue lines indicate links, so basically enzymes that are connected one metabolite to the other. But this is not all, because if you look at this model a bit more closer, you can also see there's all kinds of red lines and green lines that are connecting other parts of the network to each other. And these are also relations, enzymatic relations that exist within such a model. But because of visualization properties, you cannot really display them in a normal fashion, because this would make a big cloud of lines and dots connected to each other, of which you cannot make any sense anymore. But these relations are in these kinds of models, and you can use them for doing calculations on these models. And in this case, this example of Lactobacillus plantarum, just to give you a bit of an idea on the size of these kinds of models, this contains more than 700 genes, more than 700 reactions, and more than 500 metabolites. So it's not a small model, it's a big model, and on which you can do all these calculations. Now, how can you use these models? There's multiple ways of using these models, and I want to enlighten two of them today. One is related to data interpretation, because you can use these a lot and very useful for visualizing and interpreting your transcriptome data, because you can really now understand what's happening in your fermentation from the metabolic point of view. And secondly, you can also use these models to do a flux prediction of the reactions in your organism. So basically, what you can do is you can take different media compositions, you can add these to your model, and you can just predict how your organism will grow given the medium you supply. So with respect to the transcriptome data analysis, I want to show you one example here. This is a case which we studied in Lactobacillus plantarum, and if you look at this growth plot here, so what you see here is the OD measurement on one axis and the time on the other axis. When you look at this growth protocol here, it looks sort of normal, but what you can clearly see that there's somewhere during the logarithmic growth of the organism you see a reduction in the growth rate. Even if you would display it in a bit more different way, so just take the first order derived from this plot, you can see that indeed there's a clear drop in the growth rate of the organism. The question, of course, is what is causing this? And normally when you would do this in a lab, you would have no handles to really examine what's going on here. Just to give you a bit more information on this, so we grow it on an undefined growth medium, so from the medium point of view, we're not really clear if there may be something lacking. There's no pH control. Cells are not in respiratory mode, so they're growing anaerobically. And they have a consistent and reproducible delay in growth. So what you could do in this case, and this is shown here at the bottom of the slide, you could of course take samples during the fermentation. So in this case we took two samples, one just before and one just after the inhibition in the growth rate. And what you can take those samples and do a transcriptome analysis on them. We'll not go into detail in transcriptome data today, but basically what you get is for each gene an idea what the expression was before and after the delay in growth. And this is just visualizing this information on top of such a metabolic model. So what you can see here in yellow are all genes that were not changed in expression when comparing P1 to P2. The genes in red are all genes that were upregulated under the P2 condition. So they were low on the P1 and they were high on P2. When you look at this plot on first hand, it doesn't really tell you anything. So you have an indication something happens here. This is basically you're looking here at a part of the glycolysis. So the metabolite in the middle is pyruvate, but you're not really clear what's happening. But this model now allows you to shift your view on these reactions, for instance, by not looking at the enzymes, but you can also just look at the metabolites. And when you do that, there's something clear coming out. So this is just taking one of the metabolites, in this case CO2, as the central node in visualizing your data. And what you can see is that all the reactions that go through CO2, they're all upregulated under P2 compared to P1. So it might be, in this case, that CO2 is the main metabolite which is important in the relation of the change in growth rate. And on basis of that, we thought of the following hypothesis. So what could be the case is that CO2 is flushing out of the medium during growth. This, of course, then has as a result that growth would arrest. The organism reacts to that because it is able to produce CO2, but the reactions were not used anymore before we started, at the moment we were starting growth. So that switching on CO2 production, and by switching that on, growth resumes again, and it will grow happily further on. Now, to cut the story short, of course, what you need to do with these kinds of models, test this case in reality, and what we found was that indeed, when you would grow the cells supplemented by 1% CO2, you don't see this change in growth rate anymore, and the cells just happily grow on during the logarithmic phase. Second example I want to show you about metabolic models is more related to how to optimize your fermentation conditions in order to get more biomass. So in this case, what you can do, you can take the model and you define which components in the model are important to construct biomass. And you can just ask the model, okay, predict the situation in which case I can make most biomass given my input. So basically the model will predict all the pathways that go through your metabolic model given the input. And then the input is of course your medium composition. So the sugars, amino acids, vitamins, all the things which are normally present in your medium. And on basis of that, you can get a prediction of what the biomass would be in the most ideal case. Of course, in addition, you can also find what are the end products that this organism normally produces in the most optimal case of biomass formation. This is of course interesting, but what this system allows you in addition is that it also gives you an indication on how important these different metabolites are. So in this case, showing you an example, what it shows is for each of the compounds which you have present in your medium, it shows them how much they are used and how much is present. And in this case, this example, you can see that there's two compounds here indicated in green and in red, which are produced when it's a positive sign, or which are used by the organism, and in this case we're limiting. So the compound Y, which is here used at a level of -10 millimolar, was actually the amount that was present in the medium. So this is an indication that this compound Y is probably limiting in this specific fermentation process. That raises, of course, the hypothesis, if we would add more of compound Y to our medium, would that then increase the biomass yield? And in fact, that's shown here in this graph, it did. So when you look at the blue graph here, this is showing you the normal graph under the normal medium composition. And when you look at and when you compare that to the green graph here, this is showing you what happens when I add compound Y at a double amount present in the medium. And indeed, this has a drastic effect on the biomass produced. So here you're looking at OD 600 counts reflecting the biomass present in the medium, and you can see even that this is that there's an increase of around 1/2 times more of biomass, but just adding a simple single component to your medium. Okay, so I tried to show you today what the relevance is of these metabolic models and how you can use them to improve your processing conditions or your fermentation conditions. And I just want to spend a slide on telling you that, of course, you can do this not only for model organisms, but you can basically do this for any organism for which you have a genome sequence. So as long as you have a genome sequence, you can make a model out of it, and you can use that model to reflect the real case scenario. Of course, we also have already models available already. I just wanted to list a few of these, so if you have interest in using these kinds of models, you don't need to go through the complete process of constructing a new model, but we can just take the models that are already present. So concluding this part of the webinar, what I wanted to show you today is the use of single-strain models and how they can benefit you in your research. First of all, by helping you visualizing your data and interpreting the results that you get from the lab, which can help you in leading to a faster strain development without having to scan through millions of Excel files, but by just displaying them on your screen. Secondly, it will help you in finding leads to improve engineered strains by trying to find out what can still be improved on these strains in relation to productivity or robustness in your fermentation process. What I also tried to show you today is you can use it for medium composition and for optimizing medium composition and fermentations, generating basically higher yields and more robust fermentations. And finally, something I did not show to you today, but also things you can do, you can even use this system to create in silico knockouts. So you can just knock out the single reaction which is related to a gene and see what this effect is on the growth of your production organism without making the knockout itself. This brings me to the final topic of this webinar today, and this is related to microbial community profiling. So if you look at microbiota composition nowadays and the research that is done to it, I think it's well known that there's many different diseases for which we know that there's either a relation or a direct causative relation with microbes or microbiota composition and the display of these diseases. This is of course very interesting, and you can use that to create therapies that allows you to reduce the problems that are related to these kinds of diseases. But more and more often, people are now also showing that also health is for a large part dependent on the microbiota that you possess. And this is just showing you a few examples of papers that have come out recently that describe that, for instance, pigs that swirl around in the dirt are often less susceptible for diseases than when they are in a very sterile environment. This was related to the microbiota that they possess because they are in a more flexible environment themselves. So there's more and more information leading to that also health and being healthy can be monitored by looking at your microbiota profile. This means that there's more and more research being done related to different parts of the human body or of the animal body that are being profiled for microbiota. And just to give you a few examples of things we've worked on here at NIZO, so we've done studies already on skin. So looking at different parts of the body on the outside, so taking samples from the skin and looking at the microbiota related, for example, to psoriasis or atopic dermatitis, which can of course have a relation with, and of course relations to less severe diseases or problems like acne, dandruff, and malodor. We've also looked at oral health, so the microbiota that is around in your mouth, especially related to breath quality. So how does the microbiota affect the quality of your breath? And of course, maybe the microbiota which is most well known by most of you, the microbiota present in the GI tract, either of humans but also in pigs, fish, and for instance chicken. And there we've done really a lot of different studies relating from dietary intervention studies all the way up to microbiota and GI comfort or immune development. The means by which we study these microbiota profiles is most often done by barcoded pyrosequencing. What this basically does is you take a sample from the microbiota, which can be intestinal content or feces or a sample from the skin, and what you do is you do DNA isolation, then perform dedicated PCR to amplify a region in the genome which is basically telling you something about the diversity of the organism. So in most cases this is part of the 16S RNA gene. So you do a PCR and then you go and sequence these PCR products. And then what comes out of it is a big bunch of raw data of all the different rRNAs that you can find in the specific sample that you take. And what I want to focus on today is, well, how can you then use this information to really distill differences that you find between different individuals or between treatments, and identify the key players out of that. So what we use here at NIZO for this analysis, basically you start off with taking your raw sequences and the metadata. So the metadata basically describes what is the sample like, was it a treated or a non-treated individual, which individual was all the kind of information that's just describing the sample. And we take those two and we put them into a standardized pipeline, which is called CHIME. And I think this is the best available software for analyzing these properties. The downside, however, of CHIME is that from the downstream processing point of view, there's far better visualization and interpretation techniques available. And what we've done here at NIZO is that we've developed several of these methods which really allows you to take the results from the CHIME pipeline and interpret them to get an identification of most important changes in these microbiota. And just to show, and I would just want to spend the last few slides on showing you a few examples of this. So in this case, we're looking at an oral microbiota that was isolated from the tongue, and here we compare a healthy volunteer versus a halitosis patient. When you look at this, and what we use for this are graphs which were developed by Sundquist, which we call PhyloVis, where you can really look at the populations present in the microbiota. And the way you need to read these graphs is that basically they show you the diversity of the sample at all kinds of different levels. So all the way to the left, you see the diversity on the level of bacteria. Then when you further go down, you will get down to the phylum level, phylum, family, species, all the way down up until the level of species. What you can also see is that in this graph, when you take it from left to right, every now and then you see that there's parts of this tree that are being cut away. And this basically means that from that point of view, for this specific set of organisms, you really cannot define what the exact next level in taxonomy of these species is. For instance, you can identify that it's a bacilli, that is a member of the bacilli, but you cannot really identify whether it's a lactobacillus or a bacillus. And then what you can do with these graphs is you can apply them then to the different samples that you've taken. So here you look at the healthy volunteer, and when I take into account an additional graph here, this is showing you the profile of a halitosis patient, and then you can of course compare these with each other. And in this case, this is of course a clear-cut example. That's why I use it in this webinar. You can see the actinomycetalis and the bacilli being dominantly present in the healthy volunteer, while if you look at the halitosis patient, those species have gone, and the Prevotellacea and the Clostridialis, those are overexpressed in a halitosis patient. And this could explain part of the disease phenomenon that you see. Of course, this is a nice way of comparing the two samples, but you're not really comparing them statistically. You're only comparing them from the way of looking at them. And what you can, in addition, do, look at it in a different way. And this is a graph which also shows you the comparison between the same data sets. But in this case, each node here represents a distinct phylogenetic species or a taxonomic group. And the size of the node in this case shows you how dominant this species is, while the color of the node shows you the comparison between the two different samples you have. So in this case, when it is predominantly colored red, it means these strains or these species are more present in a halitosis patient, while if it's predominantly colored blue, they're more present in the healthy subjects. In addition, you can add all kinds of other statistical information to these plots, like in this case done by looking at the thickness of basically the surrounding of the node, which is indicating you the supportness of the observed difference. So in this case, if this phenomenon, this species was only found dominant in only a single patient with halitosis, this would be colored white, while if it's present in all halitosis patients, it will be colored black. Finally, and this is the last slide showing you an example of how to visualize these type of data, you can also do this in a more sophisticated manner in trying to identify which species or which strains or taxonomic groups are most important to distinguish between the two. In the previous slide we saw all the differences, but which of these differences is now the most important one? And we've developed a method basically applying a machine learning-like technique to identify which of the taxonomic groups is most important in discriminating. In this case, for example, between different volunteers. And just to give you an indication that this works, what you see here now on the right, in the top row, the colors blue, brown, and purple give you different samples of the same volunteer. And the red, green, and yellow coding indicates the level at which a specific taxonomic group was present or absent, present in green, absent in red, in the specific individual. And looking at this graph, you can immediately see that the first and the best discriminating one is a taxon which is found always present, for example, in the purple individual, while in the brown individual it was always absent. So when you want to distinguish one individual from the other, this specific taxonomic group, which is the Streptococcus, would be the best in discriminating the two from each other. So this information you can also not use only for healthy volunteers and comparing different volunteers, but you can also use the same analysis method, for example, to relate to dietary treatment, health status, or immunity response. Okay, summarizing the third topic of today that we've addressed in this webinar, microbial community profiling. What I wanted to show you is that you can identify using 16S pyrosequencing which microbes are present in your sample. You can look at all kinds of different microbiota. For instance, we've looked at human microbiota today, but you can of course also do this in fermented products, production plants. For instance, we're looking at biofilms formed there, or environmental samples. And it will give you an in-depth view into which species are there and how dominant are they. And especially also in cases of the level of, and we can do this on the even up to down to the level of species, depending on the primers you use. So in this case, we talked about RNA-based primers, which will give you a quite decent indication of which species or which families are there. But if you take other genes or other parts on the genome to take as a benchmark or as a part to amplify, you can even go more in depth in trying to identify more to the level of the species. So summarizing the webinar of today, I tried to enlighten 3 topics of bioinformatics to you and how they can be used basically in the broad view of food research. And I just wanted to close with this slide showing you basically the procedures or the analysis that we do at NIZO in relation to bioinformatics. So I showed you things on sequencing and annotation and all kinds of things we can do there. We can perform metabolic modeling, metagenomics, 16S pyrosequencing, but also true metagenome sequencing. And what I didn't highlight to you today, but which we're also capable of, is performing microarray or transcriptome data analysis and mining those data sets. So with that, I would like to thank everybody for their attention, and I just want to thank a few people. First of all, the complete bioinformatics team at NIZO, Sacha van Heijem, Jos Boekhorst, who's present here as well, Sege Kruiswijk, Erik Smit, and Wienand Alkema. And I would also like to especially thank Jos for well, being there and answering the questions that come from the audience, but also Maarten Hendrik from our PR department for helping me out with organizing this webinar, and René van der Meulen from our IT department for setting up the complete software package here. Next to that, I would like to thank a few of the institutes we work together with, the Kluivert Center for Industrial Fermentation, the Top Institute Food and Nutrition, and the Center of Molecular and Biomolecular Informatics, which is the Bioinformatics department at the University of Nijmegen. Again, thanks for your attention. Feel free to ask questions at this moment. If you're not able to ask any questions, or if you have any questions at a later stage, do not hesitate to send me an email to the email address posted on this slide. Okay, so this finalizes the part of the webinar. I'm now looking at Jos. Whether there are still any topics or any questions you would like to address in a plenary fashion, the floor is open to that now.
Well, there's Michiel, there's one question that might be it's a bit of an open question. So the question is, can you run by me as a novice? How can I use bioinformatics to optimize fermentations? So it's a very generic question, but what would be your take-home answer? How can I, as a novice, use bioinformatics to optimize fermentations?
So one of the things I would do is, if this is a fermentation, so I'm guessing this is a fermentation which is pretty fixed. So you have one organism and you want to produce that in a controlled fermentation. In those cases, when you have absolutely no idea on what would be well, what if you have no idea at all what the reason is why you can't optimize this organism any further, my idea would be to look at the organism in the fermentation, take samples, and first perform a microarray analysis. If you first of all do the sequencing, if you've done that and you know which genes are present, look at the activity of the genes, because it could well be that just looking at the transcriptome activity, you can find that certain metabolites are limiting in your fermentation, and this limitation is often this is giving you an indication, and this indication can be given to you by the expression of certain genes that basically are going to synthesize this lacking metabolite.
And Michiel, could we then also steer fermentation to a specific end product of interest, so a specific flavor compound that has our main focus?
I think this is possible by basically looking at, well, what are the pathways that are forming this specific metabolite. And if you know what the pathways are to form this specific metabolite, you might get an indication on basis of such a genome scale model, what would be the additions that I need to do to my medium to steer the organism in a direction of producing more of a specific compound. That would be the most natural way of doing it. You can also, of course, think of more GMO-related procedures, but I think in case of producing a flavor, which probably relates to having the organism in a fermented product, I don't think this would be the way to go. So you could think about, well, altering your medium or your raw material in such a way that the organism is steered towards producing a specific metabolite.
I think that's a good answer, Michiel. We are actually at 4 o'clock now, so there's still quite a number of questions I have in my chat window, but I think most of them deserve a more in-depth and more detailed answer than we will be able to provide now. So I suggest we get back to you specifically by email. And I think this will be a nice moment to end this webinar, Michiel. Do you agree?
I'm perfectly fine with ending. I'm also fine to just answer one or two questions, and then the people that can stay around can get some more information, and we will try to summarize and email those questions, the answer to the questions as well.
Sounds good. So let me pick.
So maybe do one or two more questions.
Okay, I'll pick one more of the questions. So this question is actually regarding how many species we can study with the different approaches. So we have talked about microbial community composition. We have looked at genome sequencing. We looked at the modeling, and of course, for every of these three topics, there's a difference in at how many species can we look in one go. Could you comment on, for these different techniques, how many species in one go do we apply them to?
Yeah, so taking it from the lowest number to the highest number, I think especially the metabolic modeling, well, this is fairly limited. So doing a metabolic model on a single organism is already very time consuming. So this is something that you can do. We have experience in the past by also using genome scale models for communities. And basically we applied it to the most simple community which we know in a food product, which is yogurt, where you have two strains that work together, and those you could use, and in that case you could basically use as you could look at the interaction between these two organisms. At the moment we are starting up sessions or tests where we try to expand this a bit further to looking at simple cheese cultures. So where you have multiple organisms up to around 5 or 6 different strains, and try to get an idea on how these basically depend on each other. Looking at genome sequencing, I guess there's no real clear limitation. But at the moment, the typical data sets we're working with vary from around 10 to 40 different organisms. I think from a cost perspective that is still doable. If you go much higher, the question of course also is, if you sequence more than 40 strains of a single species, will it add much more to the information you have on the strains? And is it worthwhile in doing that? Because actually the sky is the limit, but typically we work around with 10 to 40 species. Looking at the microbiota profiling, yeah, there of course it's not the question of how many species you can use, but how many samples can you take? Because a single microbiota sample can be as diverse as possible. It only has an effect on maybe on the depth at which you can go through this tree. But in principle, the number of strains or species or families that are in such a microbiota, that's unlimited. The number of samples you can take along, there I think we typically work with sets of around 20 to 40. And this is also because this is approximately the amount that you can fit into a single sequencing run, or that you can fit into 2 sequencing runs, and get still enough detail on the relative abundance of these strains to each other.
One thing I'd also like to add about this microbiota profiling is that, of course, as Michiel describes, sequencing-wise, the number of samples is the limit. But the specificity, so how well can I distinguish different bacteria from each other, depends on the gene target you're working on. So Michiel now mainly described the 16S sequencing, and this allows us basically to go down to the genus level. In most cases we can do some tricks to get estimates on species. But if you have a question that is basically important to zoom on a specific taxon, let's say we are only interested in bifidobacteria, or we are specifically looking at enterococci, we pick different gene targets, and using different gene targets you can basically develop your method in such a way that you can distinguish specific strains from each other. Of course this does mean you're not picking up all bacteria anymore, but only your group of interest, but that can also be an advantage. So it's a trade-off between samples, reads, but also specificity of your taxonomic classification. Okay, there was actually one short follow-up question from the audience that I think is also relevant now. And basically the question was, can we predict the impact of a microbe on a community based on its metabolic profile? So basically here we start merging these different types of analysis. So Michiel, what are your thoughts on that?
Yeah, so this basically would mean that you would need to look at what are the metabolites produced by a specific organism, and what's the relation of this organism compared to others in the microbiota profile. And I think this is still rather difficult, I would say. First of all, I think getting a metabolic profile of a single strain is probably possible. The question, of course, if you're looking at a single species, how do these different strains in the species, which strains of the species are there in your microbiota? Because this can be very variable. In our experience, the variability that you see among strains is sometimes as big as the variability that you see between species. So what the metabolic profile of a single strain is, what the effect of that is on the microbiota profile, then you would really have to be sure that this is the single strain that is causing, that is interacting with it. Of course, looking at the application of that, you could do this for dedicated strains you want to use as a probiotic. So what you could do is you could say, okay, I have this specific metabolic profile of my strain, and I have another strain which has a different metabolic profile, and I apply them both to the same microbial community. And then what you could do is you could take basically the different metabolites produced and the levels at which they are produced, and take the microbiota profiling data. So which strains are affected by this one strain with this one metabolic profile and the strain with the other metabolic profile, and then just look at which strains are affected. So what's the effect of the metabolic profile? And you could even, if you would do this for multiple strains, identify which metabolite is related to the presence and absence of a specific clade, species, phylum in my microbiota. So I think looking at from that perspective, you could, well, basically link the metabolic profile of a single strain and what the effect of that is on your microbiota.
Thank you. I think we have now basically addressed the more generic questions I've been seeing coming by in my question screen. There are still quite a number of questions that we haven't specifically addressed yet, but I think these deserve more attention than we can now give. So we will get back to you on those questions by email.
I think that's a good idea to do. So I think this will then close our webinar. Again, everybody, thank you for your attention. Looking at Jos, I think there was a lot of response coming through from you with different questions. We will try to address these questions and answer them to you on short term. And I hope this was all very useful for you. For us, I think it was also a nice and new experience. And I hope for many of the people to be in contact with you via email.